Thank you very much for the introduction. As was already said before, I'm going to give you probably a slightly different talk to many of the talks you have already seen today, and that's predominantly because I'm a computer scientist. So I work very much at the intersection between computer science and imaging, trying to actually work on the topic of deriving clinically useful information from images. And I'm going to show you in particular some work we've been doing on cardiac MRI, which might be of interest. And really the approach I want to present you today is to actually take different components of the imaging pipeline, image acquisition, image analysis, computational modeling, as well as statistical modeling, and to combine that actually with machine learning, which is one of the key ingredients. But the second thing is to also combine this with very large data sets, which we have available, and many of you might actually be familiar with. The UK is leading a study called UK Biobank, where the aim is to actually scan 100,000 people with MRI, including cardiac MRI. So what I want to do in the next couple of minutes is give you an overview of how we can use machine learning to actually improve these different aspects of the pipeline, and then actually show you some applications in trying to make, for example, association or predictions. So let me start with actually the topic of image acquisition. And it was already mentioned before that cardiac MRI is probably not always the modality of choice, because even though it provides you with excellent contrast, soft tissue contrast, as well as dynamic imaging, where you can look not only at morphology but also function, cardiac MR has one fundamental drawback: it's a relatively slow imaging technique. So cardiac MRI is not real-time like ultrasound, and therefore is often for the patient much more difficult to tolerate. They have to hold their breath, or in the case of children, of course, it's even more complicated. So I want to show you a bit how we can use machine learning to help accelerate the way how we acquire the images. And I'm not going to really explain you how cardiac MRI works. And by the way, if you wonder, it's not quite like what you see here on the slide. But the key thing to remember is that cardiac MRI acquires data in a domain called the K-space. And that's effectively the Fourier transform of the image you finally see. So we make measurements of frequencies. And the key aspect here is that we have to traverse this K-space in a sequential fashion. And that is actually what makes cardiac MRI such a slow modality, because we can't really parallelize this process. Once we have acquired the K-space, we can do a Fourier transform, reconstruct an image, and that's what you would typically look at and use for planning, diagnosis, and follow-up. So how can we accelerate this process? Well, there's a very naive approach of accelerating the process, and that is, instead of fully sampling this K-space, let me just throw some data away and randomly sample K-space. So what I'm going to do here is I'm only going to acquire a fourth of the data. Now, a fourth of the data also means immediately that you're going to accelerate the acquisition by a factor of 4. So it's obviously much faster. However, if you look at the image, you can clearly see the image is much worse and therefore much less usable. So what we are trying to do here is actually take those subsampled measurements and trying to recover the original image without sacrificing the quality of the original image. So the way we do this is based on a theory called compressed sensing, which has been around for a couple of years and is used in MRI, but we couple this also with machine learning. And the idea is we take our dynamic sequence of MR images, which is sort of visualized here as a 3D stack. We extract blocks of data from this image, which we can actually stack into long vectors in a big matrix. So this is a very big matrix. And then we start decomposing the data, the matrix, into two components. One is what we call a dictionary, and I'm going to show you an example of this dictionary in a moment. And the second matrix is a set of sparse coefficients which are supposed to describe the image in combination with the dictionary. Now, the dictionary is actually directly learned from the data, and it's learned from the patient's data, so there is no bias in this. The dictionary you can think of effectively as a visual vocabulary of these small patches which make up the image. So it's basically a set of building blocks, a limited set of building blocks, which you can combine using these coefficients to reconstruct the original image which you see. So if you do this, you get a result like this. So what you see here is a fully sampled MRI image acquired the conventional way. What you see at the bottom is the same data set from the same patient, but acquired 8 times faster, just throwing away 7/8 of the data and only keeping 1/8 of the data. If we take these measurements and we combine this with the machine learning and try to reconstruct the original data, we can effectively recover nearly perfectly the original image. So what you see here at the top is an image reconstructed from this data, and we're showing here in a color map the difference between the original data and the data which has been reconstructed from the subsample data. And as you can see, there's very little difference. So that means we are now able to acquire the images much faster than in a conventional setting, and therefore make it for patients much easier to tolerate these scans. The second example I want to show you is how we can use machine learning in image analysis to help us with interpreting the images. And that was already mentioned a couple of times earlier today, how important having models and quantitative measurements of the left ventricle and the right ventricle and the cardiovascular system are. But of course, they're very tedious to obtain. So if you look at a machine learning view of this, clearly you can see that this is not a cardiac image. But I want to just illustrate you what we're doing is, obviously you as a human observer would immediately say this image contains a cat. And therefore, if I view this as a classification problem, I would like whatever my black box in the middle is to tell me that is with a very high probability a cat, and ideally also tell me the outline of the cat in the image. Now, if you use Google or other internet-based systems, most of these systems will be able to tell you what's in the image by using something in between, which is a neural network and a particular type of neural network called a convolutional neural network, in which you train the algorithm giving it lots of examples. And during the training, you basically penalize any error the algorithm makes in this estimation. You backpropagate it. And then when you test your new image, you do a forward pass, you're doing inference, you're going forward through your model, and you're asking the machine learning algorithm to tell you what it has actually think it is. Obviously, we can do the same thing with cardiac images. We just have to change the images which we're using for training. Namely, we use MR images and we use hand-drawn segmentations by experts, and then we effectively proceed in the same manner as to what I've shown you before. And the good thing about this is a fairly generic technology, so that means actually if I change the images I'm using for training as well as the labels, I can train the system to learn different aspects. So that's a quite important step forward from handcrafted algorithms. Obviously, we're also interested in motion. Motion is a very important aspect of cardiac function, and we can try to extract motion from these type of images from conventional MR images. But we can also play a trick with the image acquisition and acquire what is called so-called tagged images. And this is here a very clever MR physics trick where you superimpose a grid of saturation lines into the image at the beginning of the cardiac cycle, and these lines move with the tissue as the tissue deforms. You can see obviously that in the blood pool these lines are immediately lost because the blood flows in a turbulent way also in and out of the plane. But the tissue maintains these lines. We can track these lines using automated techniques, and if we then combine the segmentations, as well as this motion information, we can not only segment our images over a dynamic sequence of images, but we can also map at every point on the surface. We can analyze the dynamic behavior, for example, by looking at strain values and other characteristics, wall thickening, and so on. So if we're able to do this in a single subject, we haven't actually learned very much about, for example, a population of subjects. So I'm going to talk very briefly about this idea of computational anatomy, which, by the way, in brain imaging and neuroimaging has been around for 20 years and has driven many developments which help understand the brain better using imaging. One of the key aspects, one of the key tools which we need in order to answer the question, how do we, for example, compare images and build computational models, is how do we establish correspondences. Let me give you an example. I have an ultrasound image and an MR image of the same patient, and I would like to actually integrate them and actually find correspondences between corresponding points in the image. We can do this in an automated way, and there are many algorithms now which can do this quite robustly. So I can, for example, here in this example, fuse my 3D ultrasound with my cardiac MR image in a very reliable way, and I can actually integrate the information from both modalities. However, I can also apply this to find correspondences across different subjects and different populations. So if I have a large database of images from different subjects, I can actually try to build a model finding out what is the average shape and what is, for example, the variation of the heart. And to illustrate this, I want to show you this very simple example. Let's assume I have a population of only 5 subjects. And when I mean subjects, I really talk about the images. What I can do is I can use these algorithms, these registration algorithms, to establish correspondences between every point in the image, every point in the myocardium, for example, in every subject. Once I've done this, I can then compute what is the average shape of the heart, what's the average image effectively, and I can repeat this process. And I want to show you this in an example. This is the average of 1,000 cardiac MR images, all volunteers acquired at one of our hospitals. If I don't do any registration, obviously I just see a bright blob because every patient or every subject will have their heart in a different orientation, shape, and position. If I align all the images to correct for the fact that they all have a different orientation, different size, and different position, I get an average of 1,000 heart, which looks like this. And if I actually add also some additional information to compensate for shape changes, I get an even sharper looking average image. So we have done this with, as I said before, over 1,000 subjects. We can also use, of course, segmentation information. I'll come to that in a second. And so you end up with an average model of the heart from these 1,000 subjects. And of course, we can also do this in a dynamic fashion. So we don't have to actually restrict ourselves to just a static model of the heart. And by the way, these data sets are all publicly available, so if you want to use them, you can download these. As I said before, if we add a segmentation component in, we have a mesh, an LV mesh, an RV mesh, a myocardial mesh for every subject. We can build average shape models, and if we also track the motion of the heart over over the entire cardiac cycle, we can also build what is effectively an average model of the contraction of the heart by looking at the motion pattern, the average motion pattern over our subjects. Of course, in many cases, I'm not only interested in the average, but I want to actually learn something more. And that brings us really to the statistical modeling aspect and to the last part of the talk. So as I said before, we can compute these average models of the heart. We can do this either with images, as I've shown you before, or as I've shown you also with these surface meshes. Once you do this with the surface meshes, you have effectively every heart, for every point on your surface mesh, you have established correspondences to all your 1,000 subjects. So what you can now do is you can use your favorite statistical analysis technique to look at what is, for example, the most significant variation of LV geometry or RV geometry. You can do that, for example, by looking at the principal modes of variation using a principal component analysis. And what you would end up here is a model which not only shows you the mean shape, but also shows you what is the most significant variation in the population. Obviously, the first mode is very much related to size, but it explains around 40% of what you see in the population. The second mode explains much less, and so on. So you can now describe every shape, every geometry you see using a fairly small number of parameters rather than having 40,000 points on a mesh, which are very difficult to handle. So finally, what can you do with this? What other statistical analysis can you do? This is something which was published last year by our group in collaboration with Declan O'Regan's group at Imperial College. And what we're here using is we're using a cohort of 1,200 volunteers. They're imaged with high-resolution cardiac MRI imaging. We automatically segment every image. We map all the meshes to this atlas, which now provides our unified common coordinate system. And what we can then do is for every mesh point, we can compute phenotypic features, wall thickness, for example, relative wall thickness, wall stress, and so on. We now have that for every mesh point, and we have correspondences across all mesh points. So now we can do statistical testing, for example, using regression models where we can correct for all the nuisance variables which we're not interested in, and we can then obtain, for example, in this particular case, we were interested in correlation with systolic blood pressure, and we now obtain maps like this, which show you at every point on the surface what is the strength of correlation. So red will tell you that there is a positive correlation, blue shows you negative correlation, and you can now subdivide your population according to whatever criteria you wish and produce these maps to look for differences across the population. Here's another example where we have done this in a slightly enlarged cohort where we're looking, for example, for a relationship between LV geometry and body composition, in particular lean mass and fat mass. And again, you see you can now produce these vertex-wise correlation maps, and you can also do statistical testing on the significance of these, and you can, of course, also correct for multiple comparisons, which is, of course, you're doing here 40,000 comparisons because you have 40,000 vertices, so you can't just do your traditional p-value calculation because, of course, that would mean that you probably get, if you use 5%, you would get probably 5% of the vertices showing up. Again, actually, the neuroimaging community has developed a lot of statistical tests for exactly that purpose. And here's sort of the final example I want to show you where we're trying to now go towards outcome prediction in this particular case in patients with pulmonary hypertensions. We have the same pipeline effectively. And now if we, for example, look at the 3D motion pattern, I'm afraid it's probably too small to read this, but the 3D motion patterns offer you by far the best prediction of mortality compared to all the other more traditional summary measurements you might compute for your cardiovascular system, such as ejection fraction, volumes, and so on. So looking at this more vertex-wise measurement is more accurate and also shows you larger differences. For example, if you want to stratify your groups and look at, for example, things like survival time. So I want to stop here and conclude. I wanted to show you that if you combine machine learning with image acquisition, analysis, computational anatomy, and statistical modeling, you can really make quite a lot of advances. Key ingredient here is machine learning, but the second thing is, which I haven't really touched on very much, is that there are now more and more of these large cohorts available, and also for scientists, like computer scientists and engineers like me, they're also publicly available, so you can actually share them and work with other collaborators on this. And I think combining all of this will really enable a better discovery of associations between phenotypes and clinical information, but hopefully in the long term also lead to better prediction of patient-specific outcomes. The challenge, of course, is validation, because we're already struggling to get enough example data sets to train our algorithms, our machine learning algorithms. And of course, if you want to do this in a principled way, you need to make sure that when you perform your validation, you have a completely separate data set from which you have trained on, and that's very costly in terms of resources of collecting this data. There are new machine learning algorithms which are developed, which really offer also very exciting opportunities for not only looking at imaging separately from non-imaging data, but really provide a much more integrated way and principled way of integrating imaging and non-imaging data. And finally, all of this really requires a collaboration between different disciplines, and that's actually what I find very exciting. I will just stop here with an acknowledgement of all the colleagues who have helped with the work, and of course also the funding bodies who have funded this research. Thank you very much.